Guys, in this video, let us look at Human Genome Project. So basically, the goals of Human Genome Project, DNA. So you know the human DNA, right? This is the chromosome. In the chromosome, you will have DNA, all this you know. So how many chromosomes we have? We have 46 chromosomes, right? So basically, they wanted to identify all the genes in the human DNA. So basically, you wanted to identify what? The genes, right? Human Genome Project. How many genes are they saying? Around 20,000 to 25,000, right? To 25,000 genes they wanted to identify in the human DNA. Okay, what were the other goals? To determine the sequence of 3 billion chemical base pairs that make up the human DNA. So you know the human DNA, there are in this what is called as base pairs. So they wanted to identify the sequence of these base pairs and how many base pairs there are. 3 billion base pairs, right? 3 billion base pairs. They wanted to identify the sequence of this, in exact what sequence are they coming in, like that, okay? And they wanted to store this information in the databases, and they wanted to have some tools for this data analysis, etc. Then they wanted to transfer related technologies to other sectors such as industries, address the ethical, legal and social issues that may arise from this project. Okay, so all of these were the goals of Human Genome Project. Basically, it was a 13-year project coordinated by whom? By the US Department of Energy and National Institute of Health. Okay, so during the early years, the Wellcome Trust UK became major partner. Okay, additional contributions have come in from Japan, France, Germany, China, etc. And this project is complete. It was completed in 2003. Okay, so basically, to get the complete set of genetic information of humans, that is Homo sapiens, they started this Human Genome Project. Okay, so basically, Human Genome Project, what is it? This HGP, Human Genome Project, is an international scientific research project. Okay, its goal was to determine the sequence of the chemical base pairs which make up the DNA, and is to identify these genes, okay, of the human genome. Fine. They wanted to know where physically where they are present and what function they do. All this they wanted to know about the genes, right? So when did they begin? They began in 1990, right? It ran for 13 years, obviously. So they declared it complete in 2003. Correct? So 1990 to 2003. So 1990, then 2000, 2003. Exactly 13 years, right? So it was headed by James Watson. James Watson reminds me of Sherlock Holmes, isn't it? So James Watson, they are saying. Who headed it? James Watson. Looks like the Sherlock Holmes character was John Watson, guys. So we are talking about James Watson, okay? So basically, what was the objective? To understand the genetic makeup of human species and complete map of all findings. So what did they want to find? They wanted to find the genetic makeup of human species, right? What is this? Human Genome Project, HGP, okay. Now where are they applying all this? They are applying it to study individual gene, its function, its location, right? And you can know variety of illnesses, predisposition, breast cancer, cystic fibrosis, liver disease, so many things you can write here, right? Deeper understanding into the level of molecular biology, new therapeutic procedures, right? All these nowadays they are talking about a lot of ethical or unethical procedures where you can do some, what is it called? Gene modification, right? So basically, you can also understand genetic migration, genetic drift, genetic selection, etc. So basically, what are the steps of Human Genome Project? Steps. Let's look at the steps of Human Genome Project. So basically, what they will do, they will identify pedigree, phenotype they will study. So for this phenotype, let us say height. Height is a phenotype, right? They will collect all the DNA from this pedigree, right? Then they will do the genetic mapping, then they will do the physical mapping. I'm guessing this is the physical location on the chromosome, etc. Gene mapping they will do. DNA shotgun sequencing is done. Okay, these are the steps of Human Genome Project. So any doubt you have, guys, on Human Genome Project, is this clear? What exactly are the, when did it start? 1990, right? Who? James Watson. Then it ran for 13 years. It was marked completed in 2003. They wanted to get the genetic makeup of humans, that is Homo sapiens. They wanted to determine the sequence of the chemical base pairs which make up the DNA, right? They wanted to identify all the genes, around 25,000 genes they were ready to identify, right? So they are saying they did most of it by 2003. So probably God feels so about Human Genome Project. Nice. And what does man feel about Human Genome Project? There you go. Something like that. Though the genes cannot determine whether they become president, this is just a far-fetched joke. That's all for now in Human Genome Project. Hope you have understood.